This protocol provides a simple, cost-effective way to isolate ASCs without the use of harsh enzymes or centrifugation steps, which can alter the cell phenotype and behavior. The lack of harsh steps yields less manipulated cells than those isolated using enzymes and centrifugation, minimizing questions as to whether experimental observations are an artifact of the isolation method.
This explant method can generate clinical relevant ASCs to be introduced into a patient to treat a variety of diseases or expand in the lab for further research applications. It is difficult to fully explain the missing method in words. The visual demonstration can save all the scientists the hassles of learning to break apart the tissue themselves. Similar explant methods can be used to isolate other populations of MSCs, such as those from umbilical tissues.
To mince the abdominoplasty tissue samples, first transfer the tissue to the lid of a sterile 100 mm plate. Then use a sterile needle to hold a piece of tissue in place, and use a sterile scalpel to cut the tissue into less than 1 mm pieces. Transfer the tissue to a fresh tube, and invert or shake the sample 5 to 6 times to ensure mixing of all layers. Then, with a sterile spatula, transfer 2.5 to 5 milliliters of tissue to a 100 mm vacuum gas plasma-treated tissue culture plate. Measure the volume of the sample by pouring into a fresh centrifuge tube. Next, add an equivalent volume of tissue culture media.